for the present video is degradation impurities in pharmaceuticals. So in this video we are going to see what are the degradation impurities, how these are formed, what are the mitigation strategies, and how these are evaluated. I welcome all of you for the present video, and I request you to be with the video till end so that you will get the complete understanding about this topic. As we know, the impurities are those chemical substances present into the drug substance and the formulations which make the drug substance or formulation unsafe to use. Impurities may impact the safety and efficacy of the formulation. How these if have the impact on safety? The meaning of that is the impurities are toxic in nature. That's why the drug products with impurities will be unsafe to use. And efficacy means if impurities are generated, if impurities are above certain levels, then it will compromise the potency. The impurities may be toxic, genotoxic, carcinogenic in nature, and that's why these are required to be controlled. And impurity management is very critical aspect of the drug substance development and also the formulation development. These impurities may get generated during the manufacturing or during the shelf life or storage of the drug substance or the formulation. So utmost care is required if the product is sensitive for the impurity generation. Mainly the impurities are of two types. Other types are also there, which I have explained in my earlier videos. You can go to this channel and from the playlist you will get those videos. The topic of present video is degradation impurities or degradant impurities. Mainly the impurities are classified into organic impurities, inorganic impurities, residual solvents, and other type of impurities. Out of those, if you consider the organic impurities, there are two types, process related impurities and the degradant or degradation impurities. The impurities are required to be closely monitored. The impurity generation is required to be studied in detail, and the impurities are required to be controlled to establish the storage conditions for it may be API and the formulation. Also to establish the in process and finished product specifications, storage conditions, labeling requirement, packaging system, and the stability period, expiry period, or shelf life period. There are many guidelines available like ICH guidelines, European guidelines are there, US FDA guidances are there, and the product monographs are there for the impurities. All these guidelines and monographs are there for reporting, identification, qualification, and control of the impurities as per the regulatory requirements. If the products are new, if the drug substance is new, that means if it is not in pharmacopoeial monograph, then directly the ICH guidelines can be followed. Some scientific literature, if it is available, then that can be followed. And if the monograph is there, then monograph is required to be followed. And as per the monograph, the impurities are required to be controlled. Coming to the degradant impurities, these are the impurities which are formed or generated because of the drug degradation. Degradation may take place due to many of the reasons like oxidation, hydrolysis, and many other chemical reactions are there which can lead to drug degradation. Degradants may be known or unknown type, and these are required to have specifications, that means the specific limits for their identification and qualification. So identification means the identification limit is given as per the identification threshold given in the ICH guidelines. And for identification, if the degradant is unknown, that time single maximum unknown impurity specification is required to be given. And based on to the drug substance maximum daily dose with respect to the formulation and route of administration, the identification threshold can be given. Similarly, if it crosses the identification threshold, then it is required to be identified, and then if it crosses the qualification threshold, it is required to be qualified, and as per the qualification threshold, the qualification limits are given. If these degradant impurities exceed the qualification threshold, then these are required to be qualified for their biological safety. Toxicological studies are performed for the impurity qualification. Now, why these degradant impurities are formed, or how these are the impurities generated? See, these are formed due to the API degradation mainly. The degradation of the API may happen due to storage conditions like exposure to temperature, exposure of the API or drug substance or formulation to the humidity, maybe due to the moisture gain, maybe because of the light, oxidation, or pH exposure. Different pathways are there for impurity generation. So one of those important factor is storage condition. Then microenvironmental pH in solid formulation and pH in case of liquid and parenteral formulations. Age of API or age of formulation, that is the time from where from when the batch was manufactured and where or when the batch is getting analyzed or used by the patient or used into the batch manufacturing in case of API. So that is effect of aging. Excipient interaction or incompatibility between the drug and excipient may be there in the case of formulation, and other reasons like adduct formation or certain reactions between the API and the excipient like Maillard reaction of certain amine containing drugs with lactose. Then degradant impurities. Again, we have to understand that these are required to be evaluated in API and formulations both. Drug degradation pathway study including the force degradation study, stability studies, stress studies, open pot or open exposure studies, photostability studies, hold time studies. So many type of exercises and scientific studies can be undertaken to understand the drug degradation pathway and the conditions which lead to the formation of degradant impurities. So these studies are very crucial to study the possible degradant and the sensitive conditions. If these studies are performed in detail, then the degradation pathways can be understood, and based on those degradation pathways and the sensitive conditions, the mitigation strategies can be considered. Now, if you know that the drug get degraded due to some conditions, environmental factors or storage conditions, or there is a requirement of specific packaging system or storage temperature, like some of the APIs are stored at 2 to 8 degree, some are stored at 25 degree, and some may be stored below 30 degree. So based on that, you can control the degradation impurities, or you can control the generation of degradation impurities, or you can have the mitigation strategy in your hand. You can also take precautions and do modifications in the manufacturing. If you know the impurity generation pathway, based on that, precautions and modifications can be done in manufacturing. Then proper storage conditions and handling. Protection from the environmental conditions like humidity, temperature and light. If you know that your product is light sensitive, then use sodium vapor lamp in the manufacturing and use the opaque packaging materials. If the formulation is sensitive to moisture, then work under low RH or low humidity environment during manufacturing and packaging, and protect it from moisture by certain strong packaging or container closure system. You can use desiccants also. Then packaging, container and closure system can be evaluated and finalized to control the degradation impurities. Stabilization of the formulation by the use of stabilizers, by the use of some packs or desiccants and oxygen scavengers. Many times you may find that some of the formulations are stored in SDP bottle with silica gel molecular sieve, or the combination of both can be used, and also sometimes the oxygen scavengers are used. Then use of excipients, pH modifiers, antioxidants. Some excipients have stabilization effect for the drug, and by the use of those excipients, the degradation impurities can be controlled. Use of prodrugs, salts and polymorphs. So if you come to know that some polymorphs are unstable or amorphous form of the drug is unstable, then you can go for the crystalline form. You can modify the salts, or you can work on prodrug type of approach so that the degradation impurities can be decreased and the more stable API can be prepared. You can also select some formulations, like if you know that the product is unstable in the tablet formulation but it is stable in capsule formulation, then that suitable formulation can be selected, and based on that you can have the control on the degradation impurities. Then process selection. For generic formulations, generally process selection is very important, and if you know the sensitivity of your API for degradation, so based on that you can select the appropriate procedure. For example, if the API is prone to hydrolysis due to water, you can go for non-aqueous type of granulation, or you can go for dry granulation. Then you can use the protective coatings to protect from moisture, to protect from light, and to give stabilization effect to the formulation. So there are many strategies to control the degradation impurity formation. Some of them I have enlisted here. And sometimes the some excipients can be used to stabilize the formulation. Sometimes acidic materials like citric acid is used. Sometimes basic materials like magnesium oxide, sodium bicarbonate, sodium hydroxide, or specific ratio of those excipients can be used to have a specific pH environment for the API, and by that mechanism, or by that mechanism to control the microenvironmental pH, the drug can be stabilized. So there are many approaches, many strategies based on to the API sensitivity and intrinsic stability. Intrinsic stability of the API is very crucial, and to understand that intrinsic stability, the studies like open pore study, force degradation study, mild degradation studies can be performed, and based on that you can know the sensitive conditions at which the drug get degraded, and based on that sensitive conditions and the factors you can control those factors and can stabilize your API and the formulation. See, it is not possible to make the formulation or drug substance without any impurity. Some impurities will always be there, some degradation will always be there. So it should be within the regulatory requirements and regulatory limits. I hope I have covered the degradant impurities information, and I hope you may have liked this video. The impurity and the formulation stabilizations questions are always asked into the interviews. So whenever you have time, you study those things, you watch these videos so that your knowledge will get increased about the topic and you will be having good understanding about the related topics. Thank you for watching the video. I hope you will like these type of videos so that you will have good understanding and good knowledge about these topics. Thank you.